So, in our last lecture, we discussed about the different methods of culture identifications, where I had classified the culture identification methods into two divisions. One was the conventional methods of culture identification, and the other one was the automated systems or the methods. In the conventional methods, I had described about different types of tests like the catalase test, oxidase test, urease, indole test. So, there were different tests, and I had also mentioned one special test, that was the triple sugar iron test in the conventional methods, but I didn't discuss about that triple sugar iron test there. So now it's time to discuss about the triple sugar iron test. So let's continue with the triple sugar iron test. So what is the importance of the triple sugar iron test? So the importance of the triple sugar iron test is that it is used to identify the gram negative bacteria. So specifically to differentiate between the different types of gram negative bacteria, we use this triple sugar iron tests. Okay, so first of all, let's talk about the composition of this medium in which we do this triple sugar iron test. So the composition is very important. If you know the composition of the triple sugar iron agar, then only you will be able to understand the concept behind the different types of reactions or the different types of changes that are occurring in that triple sugar iron test. So this is very important from exam point of view, from national board exams points of view, and from MCQ exams as well. So first of all, let's talk about the composition of the triple sugar iron test. The composition includes, the first of all is the triple sugars. So triple sugars, what are those triple sugars? Those are the glucose, lactose, and sucrose. And this ratio is very important from MCQ point of view. They may ask you like, what is the ratio in which the triple sugars are present in the triple sugar iron agar? So the ratio between the GLS, ratio between the GLS, that means glucose, lactose, and sucrose, is 1 is to 10 is to 10. Okay, 1 is to 10 is to 10. Glucose is one part, and lactose, sucrose, each one are contributing about 10 parts. The next part in the composition is the peptic digest of the animal tissue. This provides the necessary peptones. We will discuss about the roles of the peptones later on in this video. The next component is the phenol red. The phenol red is the indicator of the pH change. So the pH change that occurs in this in this test, that is detected by the color change of the phenol red, which acts as an indicator here. Other than that, we also have the ferric ammonium citrate, which is used as an indicator of the H2S gas production. So whenever there is production of H2S, then because of this ferric ammonium citrate, there is change in the color to black. So whenever the color changes to black, that means the H2S gas has been produced in that medium. Okay, so this ferric ammonium citrate acts as an indicator of H2S. Remember, phenol red is the indicator of the pH change, while the ferric ammonium citrate is the indicator of the H2S gas production. Other than that, we also have the sodium thiosulfate as the component of this triple sugar iron agar, and also the agar. Not but no, At last but not the least is the agar. Agar is the the most important component, or the maximum amount is contributed by the agar only. Okay, all these substances are present in limited amount, in little amount, but agar is in maximum amount in this triple sugar iron agar, in which we do the TSI test or the triple sugar iron test. So this is how the triple sugar iron agar looks. See, here we have a slant on this agar. There is a slant and there is a butt. So this part which is there at the base of the test tube is the butt, and that part which is sloping is the slant. So remember, it has got two parts. One is the slant and the other one is the butt. This is very important to discuss further about this in inference and all different parts of this test. So slant and butt, you should be familiar with these two terms. Next is, when we do the inoculation of the bacterial colony in the TSI agar, then we have to incubate also. So we incubate these test tubes at 37 degrees centigrade for 24 hours. After that, after the 24 hours passes, we have to do the interpretation. So in the interpretation, we look for three changes. What are those three changes? So those three changes are whether the fermentation of sugar has occurred or not, whether any gas has been produced or not, and whether the H2S gas production has occurred or not. So these three things should be looked for after the 24 hours has been passed. So in the fermentation of the sugar, we generally look for the change in the color. The normal color of this TSI agar is red. So whenever there is change in the color from red to yellow, that means it has got some acidic pH, or acidic pH represents that the fermentation of the sugar has occurred. That means whenever the I mean when the sugars are fermented or metabolized anaerobically, then we know that there is production of the pyruvate or the pyruvic acid, which is acidic in nature. Due to that acidic nature, the change in the color occurs. Change in the color of the phenol red occurs from red to yellow. And whenever there is change from red to yellow, that means the sugar has been fermented. Okay, we can know that the sugar has been fermented. So that is the that is how we come to know about whether the fermentation of the sugar has occurred or not, by the color change. Okay. And the gas production, so what are the gases produced? The gases may be produced are the CO2 and the O2 gases. We will see how we detect that gas production, and also the H2S gas, hydrogen sulfide gas production. We will also see how to know whether the H2S gas has been produced or not. So first of all, coming to the sugar fermentation. So we know that there in the composition of the triple sugar iron, there was three sugars: the glucose, lactose, and the sucrose. Okay. So the sugar fermentation can be classified into three types. One is the so based on the sugar fermentation, the organisms can be classified into three types. One will be those who will not be fermenting any of the sugars. They will not ferment neither glucose nor lactose nor sucrose. Okay, so those will be called as the non fermenters. And some group will be fermenting only the glucose. They will be called as only glucose fermenters. And some will be those who will be fermenting glucose along with lactose and or sucrose. Okay. So those will be called as the more than two sugar fermenters. So we will see how the change in the color occurs in all of these three conditions, or all in case of all of these three fermenters, three types of fermenters: the non fermenters, the only glucose fermenters, and the more than two sugar fermenters. So first of all, coming to the non fermenters. So in the non fermenters, we see that the we know that non-fermenters means they will not be metabolizing one of the sugars. So if they are not metabolizing any one of the sugars, that means no acid or the no pyruvic acid will be produced. That means color change will not occur. Color change will not occur. So if the color change is not occurring, that means the color of the media will be red only. Okay, it will not change to yellow. So if it is not changing to yellow, that means the reaction is alkaline. Okay, that means the reaction is alkaline. So see here, in the case of non-fermenters, we see that both the slant and the butt are alkaline, and this is written as K by K, or this is said as alkaline over alkaline reaction. We call it as alkaline over alkaline, and it is written as K by K. Okay, but that is read as alkaline over alkaline reaction. Okay. And these non-fermenters are nothing but the pseudomonas. The pseudomonas give this reaction of alkaline over alkaline in the triple sugar iron test. Okay. Next comes the only glucose fermenters. So only glucose fermenters are very special because in different time duration they give different types of taste reactions. Like in the initial few hours, they will be giving the acid over acid reaction, while in the later parts of the incubation period, like more than 18 hours, if they are incubated for more than 18 hours, then they will be giving alkaline over acid reaction. Okay. So there is slight difference between the reaction produced based on the duration of incubation. So in the initial few hours, they will be giving acidic slant and acidic butt. That means both the slant and the butt both will change the color to yellow. Okay, both will change the color to yellow. That's why we are calling it as the acidic slant and acidic butt, and it is read as acid over acid. Why is this so? This is because the glucose is utilized by the bacteria both aerobically and anaerobically initially. Okay. So they will be producing pyruvate and hence this acidic change will occur in both the slant and the butt, and this acidic change in the pH will cause both the slant and the butt to look yellow, and we will be calling it as acid over acid reaction, or the A by A. But let's see why uh, over prolonged incubation it changes to alkaline by alkaline over acid reaction. So that is a very important concept to understand why why in the later parts of the incubation of more than 18 hours, why why after more than 18 hours of incubation there is change in the reaction from alkaline over the acid, alkaline over the acid. So we see that on long term incubation all the glucose is already metabolized, and since bacteria cannot utilize sucrose and lactose, so they start utilizing the peptones in the slant, and that peptone utilization can only occur aerobically, that this cannot occur anaerobically. Okay, so when the peptones are metabolized, they produce the NH3, that means ammonia, and ammonia is alkaline in nature. So it is alkaline in nature, and since this peptone metabolism is occurring only in the slant part of that TSI agar, so the slant part of that TSI agar changes the color to the again from yellow, again it becomes red. That means there is again the alkaline reaction occurring in the slant part, but since the bacteria in the bud part, they cannot they do not have the excess of the oxygen, so they are not able to metabolize the peptones in the butt, so they are not able to produce the ammonia, and hence they they are remaining acidic. I mean, they are producing the acidic but only there is no change in the color of the butt. So it is producing the alkaline over acid reaction on incubation for prolonged period of more than 18 hours. So the example is the Shigella and Salmonella. Okay, and the next one is the more than two sugar fermenters. So if there is more than two sugar fermenters, that means the if the bacteria is metabolizing both the glucose as well as the lactose and or sucrose, then it will be metabolizing those sucroses, those sugars in the slant as well as in the bud anaerobically. So when they will be metabolizing the sugars anaerobically, they will be producing pyruvate and different types of acids. So that will be produced anaerobically in both the slant as well as in the bud. That means the acidic change will occur in both the slant and the bud. So there will be acidic slant and acidic bud. That means both the slant and the bud will become yellow in color. So it will be called as acid over acid reaction, or A by A. The second change that we want to note or we want to detect is the gas production, whether there is any gas production or not. So if there is production of the CO2 and the O2 gases, then we see that is detected by, how are we going to detect whether the gas has been produced or not? So that is detected by the splitting in the agar. If we see that the agar has splitted, or the gas has been produced in that agar, TSI agar, that means the gases are produced. And if sometimes we see that the agar is pushed upwards, okay, so there is gap produced at the base of that test tube, that means also that the gas has been produced. So these are the two indicators by which we detect whether the gas has been produced or not. So the gas producers are the example is the E. coli and the Klebsiella. These are the two gas producers. Okay, next comes the H2S production. So H2S production, hydrogen sulfide production. So how does that bacteria produce the hydrogen sulfide gas? It is not normally produced in any by any bacteria. So we see that when since this TSI agar also contains the sodium thiosulfate as the component or as the composition, so when the bacteria acts upon this sodium thiosulfate, then it produces this H2S gas, then it produces this H2S gas. But how are we going to detect this H2S gas? That detection of H2S is by change in the color, if we see that the black color change has occurred in that agar, that means the H2S gas has been produced. Okay, but the, it is of surprise that H2S gas is colored. Then how is that black color is getting produced? So H2S gas is colorless, that is known to all of us, but that black color is because of the reaction of the H2S with the ferric ammonium citrate, which is again a component of that TSI agar. So when that H2S produce their combines or reacts with the ferric ammonium citrate, that produces the ferrous sulfide, which is black in color. And due to this ferrous sulfide, this black color change occurs in that medium, and we say that the H2S gas has been produced. Now this H2S gas production is done by the salmonella. This is a example. Next we will see certain test tubes of this TSI agar, and will try to evaluate whether the will try to evaluate the reactions, and whether the gas has been produced or whether the H2S gas has been produced or not. So we will see all of those three things here. So see here in the test tube number 1, we are seeing that the both the slant and the butt, both the slant and the butt are red in color. That means it is alkaline, it is K by K. Okay, it is K by K, and there is no gap produced anywhere in this, there is no gap produced anywhere in this. So we can say that the gas is absent. Okay, the gas is absent in this test tube, plus there is no black color change seen in this test tube 1. So that means we can say that there is no H2S gas production as well. So the test tube number 1, the test tube number 1 is K by K. The test tube number 1 is K by K, gas negative. Okay, the gas is negative, and the H2S is also negative. The H2S is also negative. So you should write this here. Okay, you should write about all the test tubes in this 1 2 3, like this you have to write here what are the changes that you are seeing in all of these test tubes. Okay, first let me tell you what are the different changes in different test tubes. So coming to the test tube number 2, we are seeing that both the slant as well as the butt has changed the color to yellow color. That means the acidic reaction has occurred both in the slant as well as in the butt. So we can say this is acid by acid, this is A by A or acid over acid. Again we can see that the gaps has been produced. Can you see here the gap? Can you see here the gap? So the the agar has been pushed apart from the base of the test tube. That means the gas has been produced. So here the reaction I mean here the TSI test is A by A and gas positive, but there is no color, no black color. Okay, no black color is produced anywhere in this test tube. So H2S is negative. So test tube 2, the test tube 2 will be A by A. Okay, the test tube 2 will be A by A, then gas will be positive. Okay, gas will be positive, and then H2S will be negative. H2S will be negative. This is how we interpret the TSI test. So similarly you write all these tests in this portion of your notes. You should write the interpretation of all of these test tubes like 1 2 3 4 and all. Let's come to the test tube 3. So in the test tube 3 we can see that the red color is present in the slant, but there is yellow color in the butt. That means acidic reaction has occurred in the butt. Okay, but the alkaline reaction is still there in the slant. That's why the color of the slant is still red. So the reaction here will be K by A. The reaction in the test tube 3 will be K by A. Okay, it will be K by A, K by A. But can you see any gas in this test tube? No, there is no gap produced, or there is no elevation in the agar from the base of the test tube. So we can say that the gas is negative. So the test tube 3 is K by A, K or the alkaline over acid, gas negative, and there is no black color also, so it will be H2S negative also. So the test tube 3 is alkaline over acid, gas negative, H2S negative. Now come to the test tube number 4. So the test tube number 4 is that the slant is alkaline. See here, the slant is alkaline, and the base is acidic, or the butt is black in color. Now one should you should remember one thing, that the butt is, I mean, the this change of this ferric ammonium citrate to the ferrous sulfide is possible only when there is acidic pH. So if here the black color change has occurred, that means there must have been acidic reaction. So this will be called as K by A, because here it is pink, here it is pink, so it is alkaline slant, and the butt is black, that means there must have been some acidic pH, so it will be K by A. So the reaction in the test tube 4 is K by A, gas negative, and H2S positive, because the black color change has occurred, H2S has been produced. So here in the test tube number 4, it will be K by A and gas negative and H2S positive. What will be the change, what will be the reaction in the test tube number 5? So it will be, so here see, the change has occurred in the slant, plus there has been black color change in the butt. So we can say that it is acid over acid, that means A by A. Okay, it is acid by acid, A by A, gas is negative and H2S is positive, H2S is positive. So test tube 5 is A by A, gas negative, H2S positive. Okay, now you all tell me about the reactions of this test tube number 6 and 7 in the comments below. Okay, I will be waiting for your reply in the comment section about the test results of this test tube number 6 and the test tube number 7. Okay, so this is all about the TSI agar. I hope I would have been able to answer all your questions and explain about this TSI agar, which is very important from the MCQ point of view.